Hello, everyone, and welcome to today's webinar. Deep dive into spatial biology with MERFISH technology and the MERSCOPE Ultra platform. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Vizgen. To learn more about our sponsor, please visit vizgen.com. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. I would now like to welcome our speakers. Rob Mathis, Global Instrument Product Manager of Vizgen, and Dr. Manisha Ray, Director of Applications Development, Vizgen. For complete biographies on our speakers, please visit the biography tab at the left of your screen. Rob, Manisha, welcome. Rob, you may kick us off.
Great. Thank you for that introduction. Hi, everyone. Very excited to be chatting with you all today about MERSCOPE Ultra, Vizgen's new spatial imaging platform. I'll start us off today by introducing the platform and describing its key features. Then I'll pass it over to Dr. Manisha Ray, who will share some exciting data showcasing the performance and biological utility of the platform. Before jumping into MERSCOPE Ultra, I want to take a quick second to talk through Vizgen and its core technology. Vizgen has been enabling groundbreaking research with spatial genomics through the commercialization of MERFISH technology. MERFISH was first published in Dr. Xiaowei Zhuang's lab out of Harvard in 2015 and was first commercialized by Vizgen in 2021 with the commercial release of our flagship in situ imager, MERSCOPE. MERSCOPE achieved its global launch in 2022. We've since expanded its capabilities to address the moving needs of spatial researchers to include support for new sample types such as FFPE and expand detection capabilities to include protein code detection. In 2023, we expanded our menu by adding pre-designed panels and improved our cell segmentation and image processing pipelines. Earlier this year, in 2024, we launched our custom 1000-plex panel, helping expand researchers' gene targeting capabilities and enabling deeper cell type characterization. And for the focus of today's webinar, we're excited to introduce MERSCOPE Ultra, Vizgen's latest model in situ spatial imaging platform. Vizgen remains committed to innovation and will also be launching our next generation chemistry towards the end of this year as well to improve the robust detection of MERFISH in challenging sample types. Briefly, let's talk through what is MERFISH. MERFISH stands for multiplexed, error-robust, fluorescent in situ hybridization. MERFISH is based on the same general principle as single molecule FISH, but leverages combinatorial labeling, sequential imaging, and error-robust barcoding to enable highly multiplex RNA imaging. With combinatorial labeling, each gene in the panel is assigned a unique binary barcode. Sequential rounds of hybridization and imaging are used to generate the barcode on the transcript level, with transcripts appearing either on or off, representing a 1 or 0 in the barcode. The barcode is generated over several rounds of imaging and then tied back to the original gene ID from the panel. In terms of error robustness, barcode sequences are designed to tolerate a certain number of misread bits before a barcode is misidentified as a different gene. If a readout error does occur, the system can assign a readout to the nearest correct barcode, producing accurate and consistent results. And for today, we're very excited to introduce you to Vizgen's latest model imaging platform, MERSCOPE Ultra. MERSCOPE Ultra offers a leading combination of imageable area, throughput, and sensitivity designed to rapidly generate high-quality spatial data and answer biology's most complex questions. Ultra is Vizgen's second-generation platform and leverages 3 years of on-market experience with our first-generation platform, MERSCOPE, to deliver a new platform designed to meet the evolving needs of spatial researchers across a spectrum of disease areas and applications. We are eagerly anticipating the commercial launch of the platform before the end of the year. Similar to its predecessor, MERSCOPE Ultra is a full end-to-end solution facilitating MERFISH measurements from custom panel design through data visualization and analysis. The process starts with users leveraging our interactive gene panel portal to design custom gene panels targeting their research interests. Once designed, the panel is prepared and provided in an easy, ready-to-use format. The panel, along with supporting reagents and consumables, are used to prepare the sample for imaging at the bench. Once loaded onto the instrument will automate sequential rounds of hybridization and imaging to provide biologically meaningful output in a variety of file formats to be studied directly by the user or fed into downstream analysis tools. Lastly, our visualization software lets users interactively explore their data output within the spatial context of the tissue. MERSCOPE Ultra combines the advantages of our first-generation MERSCOPE platform with significant improvements to throughput and flexibility. Powered by MERFISH, MERSCOPE offers researchers high-resolution, high-sensitivity spatial data for up to 1,000 genes in a single experiment. Custom panels allow researchers to study targeted questions across a variety of applications and species. MERSCOPE Ultra will provide the same high-resolution, high-quality data as MERSCOPE, but on a significantly larger scale. MERSCOPE Ultra offers all the above benefits from MERSCOPE, but with significant improvements to throughput and flexibility. MERSCOPE Ultra is the only spatial imaging platform with an interchangeable flow chamber design intended to maximize flexibility and reagent efficiency. It offers an expansive imageable area of up to 3 square centimeters, triple that of the prior generation platform. With advanced hardware and sophisticated optics design, the system can measure up to 9 square centimeters of tissue per week. Modern, high-performance computing power enables efficient data processing and compression to improve data portability and reduce storage costs. So we mentioned Ultra introduces an interchangeable flow chamber design, and there are 2 different flow chambers the system supports, both our large flow chamber and our standard flow chamber. Our large flow chamber is shown here and is called FCXL. With the large flow chamber, it offers an expansive imaging area that enables streamlined analysis of larger samples and helps reduce cost per sample by running multiple tissues on a single slide. The large flow chamber offers a maximum imageable area of up to 3 square centimeters. It's supported by a large rectangular MERSCOPE slide, whose rectangular tissue placement area is ideal for running larger samples, reducing the need for scoring or separating them into multiple slides, or for running multiple smaller samples in a single experiment, greatly reducing the cost per sample and also the hands-on time by combining what would be multiple experiments previously now into a single experiment on a single slide. Slide markings on the slide help users visualize the imageable area and provide guidance for tissue placement during sectioning. The marked area exceeds the full 3 centimeter area, providing some flexibility in tissue placement and orientation to help users utilize the full 3 centimeter squared area every run. The second flow chamber is called our standard flow chamber, named FCXS. What FCXS enables is the users to scale down imaging area to conserve reagent when a larger area is not needed for their sample they're currently studying. The standard flow chamber supports a max imaging area of up to 1.25 square centimeters and is supported by our 40 millimeter round standard MERSCOPE slide. Given the smaller tissue area being imaged, the reagent volume required to process these samples is less compared to that of the large flow chamber. Naturally, the smaller area on the slide is ideal for smaller sample types or for initiating optimization of new tissues before scaling up to max production with the large flow chamber. Swapping between flow chambers is easy and requires minimal setup by the users, and both the standard and large slides follow the same simple prep workflow at the bench. So more data per experiment is great, but it needs to be produced in a reasonable amount of time. And Ultra's advanced optomechanical design and high performance processing power support fast image acquisition and data processing. Per week, Ultra can measure up to 9 square centimeters of tissue utilizing the large flow chamber, or 6.25 square centimeters of tissue using the standard flow chamber. The system includes modern high-performance processing technology, including dedicated graphics processing, which enables a couple key features for users. First, it enables high-fidelity image compression, which for these large sample images helps dramatically to improve data portability and reduce data storage costs. The dedicated processing power also helps with efficient analysis to help keep pace with imaging, so the system is ready for your next sample when you are. The system also includes high-capacity storage on both the control and analysis computers with a whopping 58 terabyte storage capacity. Combined with our high-fidelity compression, this gives users the room to store up to 10 or more experiments, providing them time and flexibility to transfer data off the system when it's convenient for them. It's important to note that the improved throughput of Ultra does not come with a compromise to high-resolution, high-quality MERFISH data, and that's thanks to our system's high-performance optical design. MERSCOPE Ultra leverages an oil immersion, high numerical aperture objective. This objective, together with our MERSCOPE slides, which are designed for high-resolution imaging, resolve fluorescent transcript signals as distinct, compact spots. These small spot sizes reduce the chance for overlap, increasing information density and improving detection accuracy, helping researchers finally characterize differences between cells and identifying subtle variations between cell types and cell states. The data output from MERSCOPE Ultra comes in a variety of different file formats and extensions. First, the system provides a list of all detected transcripts and their spatial locations in 3 dimensions. This data is provided in a CSV and efficiently packed Parquet file. Mosaic images are provided for review in TIFF format. And then in terms of output from our cell segmentation pipeline, transcripts per cell matrix, cell metadata, and cell boundaries are provided in CSV and Parquet formats. All data output here is compatible with single-cell expression analysis software such as Seurat and ScanPy for further downstream analysis. One new output that we introduced with Ultra is our cell clustering annotations file that's available in CSV and high-density H5AD. This H5AD file is ready for use in standard single-cell ScanPy pipelines and helps users perform relevant analyses such as PCA, UMAP, Leiden clustering, differential expression analysis, as well as neighborhood analysis. And with that, I'll pass it over to you, Manisha.
Thanks, Rob. I'm Manisha Ray. I'm the Director of Application Development at Vizgen, and I'm going to go through how the MERSCOPE Ultra features enable a new type of biological discovery. MERFISH has been around for a number of years now. It was first established in 2015 and in an academic lab and was the first technology commercialized. Due to its history, it has the largest set of publications associated with spatial transcriptomics to date. We have over 200 publications demonstrating the capabilities of MERFISH to explore new questions in spatial transcriptomics from over 100 different institutions, both in academia and in industry. And today I'm going to describe how the expanded capabilities of the MERSCOPE Ultra allow us to take all those advantages of MERFISH and apply them to a new set of biological questions. With the MERSCOPE Ultra, we now have the ability to image up to 3 square centimeters of tissue. This is 3 times larger than the imageable area of the original MERSCOPE, which was about 1 centimeter squared. A lot of the work we've done in developing this system is enabling the capability of this larger image area without any loss of quality and speed. So here we're showing the quality comparison in terms of counts between the MERSCOPE and the MERSCOPE Ultra, both using the standard round slides and the new larger slides. In the top experiment, we're showing the results in terms of counts per micron squared, for 100 micron squared, for 13 FFP mouse livers, which are shown on the right. These 13 mouse livers were imaged in 3 centimeters squared and represent over 2.4 million cells and 1.5 billion transcripts. So it's a huge amount of data represented here. And that huge amount of data in terms of, when normalized for imaging area, the number of counts on the MERSCOPE Ultra, both with the round and large slides, is comparable to that of the original MERSCOPE. We saw the same results in the lower left panel using FFP mouse brain. And 3 data sets we're showing here cover a range of panel sizes from about 300 to 1,000 genes. So with a wide range of gene panels, we're seeing very high quality data from both the MERSCOPE and the MERSCOPE Ultra. We dig into that a little bit more and not only look at correlation in counts on the left panel, but correlation to bulk RNA-seq, which is shown in the middle. So what we're showing here is that the same experiment run on the MERSCOPE and the MERSCOPE Ultra gives extremely high correlation in terms of the total counts, which you can see for FFP mouse liver, mouse brain, and human liver cancer on the left. And in the middle, you can see for the correlation of the MERFISH data with bulk RNA-seq with the MERSCOPE data in blue and the MERSCOPE Ultra data in orange. And what both of these graphs are telling us is that we're getting very, very equivalent data between the two imaging systems. And the inset shows some examples of specific correlations between the MERSCOPE Ultra data and the MERSCOPE data. In addition to ensuring that the data was of comparable quality on the MERSCOPE Ultra, even with the expanded imageable area, we also ensured that that quality held up for both the round slides and the new larger square slides. So on the right-hand side, we show the correlation in counts between the round slides and the square slides, as well as the correlation in with bulk RNA-seq between MERFISH and bulk RNA-seq for the same 3 sample types: FFP mouse liver, FF mouse brain, and FFPE human liver cancer. And in all cases, we're seeing the same excellent performance from the MERSCOPE Ultra, and all configurations of the MERSCOPE Ultra. So you can be sure when you do these higher imageable area experiments that you're getting equivalently excellent data quality. In any spatial transcriptomic experiment, you're not just looking for what your total counts are, but you're trying to get to a biological insight. So here we dive a little bit deeper into data from mouse brain run on both MERSCOPE and MERSCOPE Ultra. We took the MERFISH data from the mouse brains and compared it with reference single cell data to establish cell types. And here we note that the predicted cell types from both data sets were comparable, and the proportion of cells of each of those cell types were comparable. We see that in terms of a pie chart on the left, as well as the comparison of the predicted cell type on both data sets to that of the reference cell type. So we're seeing really high correlation between the cell type that was predicted from the MERFISH data to that of what the cell types are predicted to be from mouse brain. So again, you can have confidence in your MERSCOPE Ultra data in terms of getting to the correct biological conclusion from your spatial data. We've demonstrated the capabilities of the MERSCOPE Ultra on a large number of samples. So far, much of the data I've showed you has been from mouse brain or mouse liver, but we've tested this on a range of samples, including fresh frozen and FFPE sample types, mouse as well as human, and human normal samples and human disease samples. And in all cases, we see the same high quality data we would expect from the MERFISH and MERSCOPE system, we see the same quality on the Ultra as well. One of the most significant advantages of the MERSCOPE Ultra system is the ability to do multiple experiments in one slide. So avoiding batch effects between, for example, multiple replicates or multiple samples from different tissue types, different disease conditions, and so on. Here we're demonstrating that with 4 adult mouse brains placed on a single large MERSCOPE slide. In this case, we placed each of those samples serially, and to confirm that there were no quality issues, we then looked at the pairalized correlation in gene expression for each of the combinations of the individual mouse brains. And we see in all cases, the correlations are excellent. I believe the lowest number we saw was approximately 0.997, indicating we're not seeing any loss in data quantity or quality with placing multiple sections on the slide. In addition to the quantitative interpretation of counts from each of these 4 mouse brains, we're also seeing consistent gene expression profiles, indicating that mouse brain 4 is giving you the same composition of cell types as mouse brain 1, so you can be confident in the consistency of the data you get from putting multiple sections on a single slide. And now I'm going to go through an example of how the MERSCOPE Ultra can help you to do multiple experiments in one MERSCOPE slide, specifically focusing on Alzheimer's disease. So this work was done by Renchao Ten, a senior scientist at Vizgen and a neuroscience expert at the company. So what we did here was we took a single large slide and sectioned normal brain and the brain of Alzheimer's disease affected patient onto the same MERSCOPE slide. Importantly, the section from just the Alzheimer's brain was quite large. Just the Alzheimer's disease affected brain would not have fit on a standard MERSCOPE slide. And in this, by having this larger imageable area, we're able to look at both conditions in the same experiment with no concern for batch effects. We took that MERSCOPE data or MERSCOPE slide and ran it against 1,000 genes of a neurodegeneration panel, generating transcript profiles. But we also were able to do protein staining simultaneously with those 1,000 gene targets so that we have a true multiomic profile of both the normal and the Alzheimer's brain, again, in the same experiment. We can use that data to do standard single cell type analyses from both the gene expression point of view as well as the spatial point of view. And we can also look at the protein staining to determine the cell composition of cells that are the cell type and the proportion of cells at varying distance from disease phenotypes present in the brain. So one of the most interesting aspects of this experiment is that the gene panel we used covered both cell typing genes as well as neurodegeneration genes. So with our 1,000-plex panel, with one probe set, we can cell type all of the neurons as well as subtypes of each neuron using the approximately 500 genes aimed for cell typing, as well as looking at the disease effects of Alzheimer's in terms of the actual disease function and the pathways that may be affected. So having the access to a 1,000-plex panel gives us the capability to study both of these biological questions in the same experiment. Combining that with the larger imageable area of the MERSCOPE Ultra means we can look at 2 biological questions on 2 different sample conditions within one experiment. The results of that experiment are shown here. We looked at the gene expression profile using the MERFISH data to assign cell types to both the normal and the Alzheimer's disease brain. On the upper right panel, we see a dot plot of the top genes expressed in each lighting cluster of the data. And this enabled us to assign cell types to each of our lighting clusters. So what you're seeing on the right is the result of that analysis. We're seeing both the spatial projection and the UMAP projection of the top 6 or so gene expression clusters. And what we see is the expected cell types were identified, and they were found in their expected patterns spatially within both brain tissues. If we can dig into that a little bit more detail and look at a few specific cell types, we're seeing the glutamatergic neuron in sort of the outer layer of the brain, where we expect to find it more in the gray matter type part of the brain. And we're seeing oligodendrocyte gene expression in the deeper part of the brain, where you would expect to see more of the white matter. So the gene expression profiles for both the normal and the Alzheimer's diseased brain are matching what we would expect from existing biological knowledge. We combined the MERFISH data with protein staining for A beta plaques, the most classical phenotype of Alzheimer's disease. So on the upper left panel, we're showing the gene expression of several neuronal markers. And in the middle plaque, we're looking at the protein expression of the A beta plaques. And you can see very clearly, while there's some background staining in the normal brain on the left side, the A beta expression is significantly higher in the Alzheimer's disease brain. And this is exactly what we would expect. If we zoom in a bit more, you can see the individual plaques and the cells associated with them. The plaques are in green, and the other colors are the different cells that are neighboring those plaques. Because we have all this information spatially, we can do analysis of the composition of the cells that are interacting with the A beta plaques, which can give us more insight into the disease progression and the disease mechanism. So the way we did this was we basically segmented the plaques to assign coordinates to them. And then we can use those coordinates to look at cells in different distances from the A beta plaques. Here we're showing the differentially expressed genes between the normal and diseased human brain, where we're looking at relative gene expression in Alzheimer's disease relative to normal. And we can see a significant change in gene expression for the glutamatergic neurons in particular, but also in all of the cell types we expect. Within each cell type, we can look for pathways that are most affected. In this case in particular, we looked at oligodendrocytes and identify what, in terms of the mechanism, may be occurring with these gene families. We can also use that data to look at the cell types that are present closest to the plaque and those further away from the plaque. So what we're looking at here is the proportion of each of the 7 or so cell types binned by their distance from the nearest plaque. And if we just look at cell number by distance, it looks like the oligodendrocytes and glutamatergic neurons are the most affected. However, when we normalize that data and look at the cell composition by distance, we actually see that the oligodendrocytes are the biggest proportion of cells affected by the plaque. And this is the type of analysis that you can do by combining both the single cell level gene expression data with the multiomic capabilities of also looking at protein data and spatial information. So by combining both disease conditions, normal versus control, with a larger panel that can do both cell typing and neurodegeneration analysis, we can have a lot of interesting new insights in terms of what these plaques in Alzheimer's disease are doing in the brain. What cell types are they interacting with? What cell types are they changing the gene expression of? And what does that mean in terms of the downstream pathways? So hopefully that's given you some insight into the interesting types of experiments you can do now with this expanded imageable area, and how doing them, having a larger imageable area, enables you to answer these questions much more quickly by having all of the experimental conditions present in one slide, in one experiment. So with that, I will wrap it up and summarize what we've covered today. Today we've introduced the MERSCOPE Ultra, our newest MERSCOPE, which expands the imageable area to up to 3 centimeters squared without any compromises in data quality. We can use that larger imageable area to do larger scale experiments, such as putting multiple sections on the same slide. And we show how putting that the enabling capability to do that allows us to ask even more interesting biological questions, such as in this example, how A beta plaques interact with the cells around them. In addition to expanding the imageable area and keeping consistent quality with the MERSCOPE Ultra, we're further expanding the capabilities of MERFISH with upcoming launch of what we're calling MERFISH 2.0, an optimized MERFISH chemistry that will improve the performance of MERFISH in low quality tissue specifically. And in addition to having these larger 1,000-plex panels that can enable you to look at multiple questions in the same experiment, we are going to soon be coming out with pre-designed panels that also have a section of the panel dedicated to custom genes. So we will be launching several panels where the majority of the genes of interest have been chosen. But if there are specific genes you're interested in for your particular experiment, you can add those in as well, adding significant ability to customize your panels to your specific biological question. So with that, I would like to thank everybody for joining us today and open it up for questions.
Manisha, Rob, thank you so much for that excellent presentation. It is now time for our live Q&A portion of our webinar. So to our audience, if you haven't submitted any questions yet, which we already have a lot coming in, please do so now. Just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. Okay, so I'm going to dive in here. Let's just start with this first one. What is included with the MERSCOPE Ultra platform?
Great, thank you for the question. So the MERSCOPE Ultra platform comes with the control box and imaging box, which is required to control the system and image your samples, along with those main components. The system does also include both the standard and large size flow chamber, giving users the ability to flex between a 1.25 and a 3 square centimeter imaging area. Also with the system, you're providing a analysis PC, again, which is to support the analysis of the large image files and spitting out a number of different analyses for users to look at and review their data.
Thanks, Rob. Next question. Is there any major difference between the workflow of MERSCOPE and the MERSCOPE Ultra systems?
Yeah, I can take that one. There's really no significant difference in workflow. The steps of the workflow are all the same. The only difference is the volume is larger for the larger slides. If you're using the standard slides, there's really no change to the workflow at all. We have updated the software, both in the on-instrument software and the visualizer. So you'll get a slightly different experience there, but no changes to the workflow.
Thank you. How long will it take to run a maximum experiment on Ultra system with largest imaging area, and what size data output could we expect?
Sure. Again, thank you for the question. So the imaging time and analysis time depends on a couple of different factors. So first, as the question hints on, is the imageable area. So what size tissue are you planning to image? It also depends on the size of the gene panel you're running as part of the experiment. So it could be 140, 300, 500, or 1,000. Beyond that, whether or not you're including sequential staining for additional genes or protein co-detection, all those configuration steps have impact on the imaging and analysis time. If we look at the maximum runtime, so that would be a 3 centimeter area with a 1,000 plex panel with full protein co-detection. That would take about 65 hours or so to image. And then in terms of data size, thanks to our high fidelity compression, it would shrink that data size down to about 6 or 7 terabytes.
Thank you. Now, of course, you know, questions are always going to come up about cost. Is the cost per run or sample the same on both MERSCOPes? Or is an Ultra run even cheaper if you compare cost per sample area?
Yep. So if we compare the standard flow chamber from Ultra to the normal flow chamber on MERSCOPE, the cost per experiment is the same. Where users will start to see differences when they run the large flow chamber. So as Nisha mentioned, due to the larger volume requirements with the larger tissue area to be processed, the total cost per experiment will be a little bit higher when running a large flow chamber. But if you break that down to cost per square centimeter imaging area, the large flow chamber is actually about 50% cheaper relative to the standard flow chamber.
Thank you. Next question. What cell types are most associated with the A beta plaques?
So we see a few different cell types associated with plaques. If we go to slide 24, we have a list of cell types associated with the different plaques. And we found that microglia and oligodendrocytes had a lot of the biggest changes in gene expression when they were next to the plaques. Some of the glutamatergic neurons were also affected. So we do see pretty strong dependence of changes in cell types with association with plaques, which again is something you could only see with a spatial transcriptomic technology.
We can't hear you, Christy.
Apologies. How many genes can we put in a custom panel for monkey samples? Is there a difference between MERSCOPE and MERSCOPE Ultra?
Sure. We can do custom design panels of up to 1,000 plex. As we noted, we will soon have the ability to do pre-designed panels with custom spike-ins, but currently, today, we can custom design really for any species, including non-human and mouse species. You may need a little more consultation with our probe design team to work with those non sort of canonical species, but we offer a lot of flexibility in terms of that. And again, we can go from 140-ish up to 1,000.
Can we design custom panels for any species, non-human or mouse species?
Yeah. So that's one of the real advantages of MERFISH. Anything with a genome, we can figure out a way to design a probe panel to it.
Excellent. Thank you. We have so many great questions coming in. I want to squeeze as many as we can during our time allotment here. Is there any backward compatibility with the original MERSCOPE platform, or do you have to get the new system, new probes, etc.?
Yeah. So MERSCOPE Ultra is a brand new platform, but all the existing gene panels, reagents, sample prep kits that are currently available on MERSCOPE today are directly compatible with MERSCOPE Ultra. MERSCOPE Ultra does introduce a new consumable. We're introducing a large format gene imaging kit that's required for use with the large flow chamber. But beyond that new gene imaging kit, all reagents are compatible across both platforms.
Will the new instrument be the same size as the previous one?
Yep, great question. So the footprint for MERSCOPE Ultra is the same footprint as the current platform. So it's still nice, compact benchtop instrument, just 3 times the capability in terms of imageable area.
Okay. Next question. I see that only brain samples are FF, but everything else was FFPE. Would you say that FFPE have been proven to be the safest sample type?
So we did do a range of FF and FFPE samples from a variety of different sample types. I don't know that I would say one is safer than the other. I think it's more dependent on the upstream experimental question. For brain specifically, it's typically fresh frozen. So that's what most archival brain samples are, especially for human brain. So the majority of the brain samples we looked at were fresh frozen. For other cell types, more of the cancer cell types, typically FFPE is what's the archival format. So There are huge banks of FFPE tissue for studying tumor samples. So I wouldn't say that one is safer than the other, but we have demonstrated that we get excellent performance in both sample types in a variety of tissues.
Very good. Thank you. And it looks like we are at time right now. I want to give both of you an opportunity to give us some final comments.
Great, maybe I'll start. So thank you everyone for joining today. Excited to introduce MERSCOPE Ultra to you guys. I look forward to hearing more feedback and stay tuned for updates as we get closer to commercial availability.
And yeah, we're really excited to provide this new product that can enable you to do much larger experiments and to be able to answer your questions without spending quite as much time on experimentation and expanding the literal scope of what can be done in spatial transcriptomics. Thanks everyone for taking the time to join us today.
Manisha, Rob, thank you both for taking the time to share with us today, as we deep dive into spatial biology with MERFISH technology and the MERSCOPE Ultra platform. I also want to thank our audience for joining us and for their interesting questions we do not have time to answer today. And additionally, those that are submitted during the on-demand period, they will be addressed by our speakers via the email address you provided at the time of registration. Today's webcast can be viewed on demand. So if you missed anything, come back, rewatch it. LabRoots will alert you via email when it is available for replay. And we encourage you to share that with your colleagues and your friends who may have missed today's live event. That's all for now. Thank you for joining us. Until next time, have a beautiful day.
Thank you.
Thanks.
Bye.